to the Weekly Bioanalysis, a KCAS Bio podcast.
Hello, and welcome to the 101st episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 45 years. My name is Dominic Guarino, and I've been in the LBA and biomarker space for around 30 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for around 30 years too.
John and I are members of our growing scientific services team here at KCAS Bio. Either or both of us are available to answer any questions you may have about our services or this podcast or any other topics of interest that you may have. We are thrilled to have you listening to the 101st episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of our latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for new topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we're looking forward to a fun episode. To kick us off for podcast 101, John is going to go over today's podcast topics a bit before we jump into the news and resources. John?
Yes, today we thought we'd look at trends in bioanalysis, evolution of client needs, instrumentation platforms, and regulatory requirements. So it's what we're doing to adapt to the changing state of bioanalysis and the problems our clients send our way.
Yeah, great topic, John. We're exciting podcast to go over, but why don't you get us started with the news and notes first?
Well, first of all, you never guess what. We're back to the GLP-1s. This one, GLP-1s may combat depression through a mood-busting gut microbe. So obviously we talk about GLP-1s a lot simply because they're dominating the market in terms of best-selling drugs, etc. And there's a lot of discussion about different indications. There's been a lot of question marks about them. In some, they seem to have some people seem to have antidepressant effects, while others, they've sort of thought they might be linked to worse mental health, but it's not clear now. In actual fact, as the article states, the sort of negative mental health was on labels and has been asked to be removed. But this is a team of researchers in China who basically found that mice treated with GLP-1s, liraglutide and semaglutide, saw Lactobacillus delbrueckii bacteria flourish in their guts, with the microbes churning out a compound that alleviates symptoms of depression. So it's sort of showing a mechanism whereby they might actually help depression. And although it's like you'd think this should be the brain, it's all about gut health, and in some of the precursor, they looked at blood from patients with major depressive disorder, and they found that natural levels of GLP-1 were lower with those in depression. When they explored further by injecting liraglutide into depressed mice, they found that peptide built up in the intestine rather than the brain, and that's what pushed them to say, well, what's going on? Looked at the actually mice feces and found higher levels of this Lactobacillus delbrueckii, and it basically that bacterium that bacterium basically supports the synthesis of cannabis-like molecule called 2-arachidonoglycerol, or 2-AG, which has a soothing effect on the brain. I'll pass it back to you because I think I'm done with the pronunciations for a while.
Yeah, no, it's become gut health seems to be all the rage, John. Right? It's
sure.
It's something where I think it makes a lot of sense on the surface, but no one thinks about, hey, is my gut healthy? And if I'm not healthy, one of the last things people are going to think about is like their stomach. Although I think now it's becoming much more aware of just how important your gut health is. I mean, it's like its own immune system. It's a very large portion of your body. And so it kind of makes sense that if you're got some bacteria going on somewhere, it could be causing depression or pain or whatever it might be. But it's a fascinating story. These GLP-1s continue to be like magic drugs, right?
Yeah,
Like not only are they able to lose weight, they're now like potentially, because John, if I'm reading it right, they removed a warning label, right? I don't know if you want to talk about that, but
I
it might
did
be the
mention it.
opposite.
Yeah. It's been requested to remove the warning label. I think it was the FDA drove that, and then actually both Lilly is definitely looking at one of their GLP. It's actually a dual GLP-1 GIP, brenipatide.
Mhm.
Administered with antidepressant therapy in terms of helping adults with major depressive disorder, and Novo Nordisk are similarly looking at semaglutide for major depressive disorder. So it is something that the lead manufacturers are looking at. How can these molecules help? So I thought it's fascinating. We've talked about this, and yet again, this may be another indication of where these drugs can help. I mean, I think it's interesting that they're talking about this and they're saying the pathway has nothing to do with the GLP-1 receptor, which may explain why different studies have come to a different conclusion about the effect GLP-1 drugs have on mental health. GLP-1 analogs can activate central GLP-1 receptors in the brain regions involved in stress and anxiety, and so the net effect on mood may depend on the balance between the opposing mechanisms of the what's what's affecting the GLP-1 receptor versus your gut health piece. And like I say, it's fascinating, and,
Yeah.
I obviously I watch these drugs with interest because just seeing the different varieties of effects that we weren't thinking of in the first place, and I mean, no one was thinking obesity when these were first developed. It was, what can we do for diabetes, and this is all downstream. Oh, we found this, and we found this,
Yeah.
and we found this, and so it's like an evolving understanding of what these drugs may possibly contribute to us, and they're fascinating.
Well, I mean, it's the label. So these GLP-1 labels had a potential warning around
Yep,
depression and
yep,
suicide, right? Now it's become like 180, right? Like now they're actually saying that this brentuximab, right, the dual GLP-1 GIP from Lilly, that it co-administered with antidepressant may help prevent depression, right? So it's like
yeah,
come full
yeah,
circle, and it's just fascinating to think about how warning labels change. As we're beginning to learn more about GLP-1s, they're helping people with late weight loss. Now there's this potential benefit for, you know, um, uh, antidepressant. I feel like it's still to be determined just how
oh sure,
powerful these drugs
absolutely,
are. I mean, I think there's some things that talk about maybe helping with remedy um, uh, migraines and things like that. So it makes sense now when you think about how hard it is to tell how effective and what it's from if someone's gut health is different. But
yeah,
gut health is playing a factor here, and I think in all of us, I think that we can universally agree to. It's just the fact that gut health, that's a hard thing to study, right?
yeah,
Understanding everybody's gut is going to be different, and then think about the research behind this, John, where they kind of took this lactobacillus kind of bacteria, and now they're starting to it's really great basic research, right, John? That's some basic research in mice, and they're finding out that, oh wow, this excess bacteria might be something that is causing some sort of depression, and lo and behold, the GLP-1s knock it down, right? And so it's unbelievable how impactful they are. And maybe just in closing this out, it's still like all this work's done in male mice, right, John? I think that's
yes, and
important.
so they don't know in female. Yeah, it is worth emphasising this is just in mice, so it's still in the early stages of seeing if this even has potential for people. But
Yeah.
again, it's identifying a potential mechanism where it might have applicability to people in the long term.
It's yet another story about GLP-1, John. I got a feeling it won't be the last,
Oh
right?
no,
I mean, I
absolutely
think we're
not.
going to
No, no.
constantly. Yeah.
And I wasn't really really focusing too much on the label change. But like I said, that request did come from the FDA because there was an analysis at the agency found no connection between the medicines and suicidality. So that's why there was the change there. I'm not here. I'm not certainly not expounding GLP-1s as miracle medicines, but it is fascinating where they might be able to contribute and where it generally help people.
I think that story is just beginning, not ending, right, John?
Exactly. Yes.
Speaking of stories, tell us about the next.
Let's move on. This is actually, again very interesting. This is. I suppose this is addressing a much older issue. This is actually new antibodies stop measles from stabbing cells. Study finds. And although we've had a measles vaccine for 60 years, much still remains unknown about how exactly the shot generates protection against the virus. Scientists actually looked at the immune system of vaccinated people to identify antibodies. It was looking at the blood of 20 people previously given the MMR vaccine, and they identified 4 antibodies that cling to the measles virus surface and prevent it from infecting cells. So the potential. So she said that was real. She said that was all real. Sorry, Sapphire. So Erica Ullman Sapphire is a structural biologist at La Jolla Institute for Immunology who led the study, and she basically said finding these antibodies was easy because measles doesn't have as thick a protective layer of sugar molecules around it as other viruses do. So it was, I think, easier to pick these apart. But although we've had this vaccine for a long time, there are actually populations such as the immunocompromised and infants who can't take it. So actually having these antibodies available as a prophylactic may actually give another mechanism to help people where there is, if a measles infection takes place. So it's interesting because the article states they never really looked into the mechanism because they came up with the vaccine a long time ago, where there wasn't the ability to look into the mechanism. Our tools have advanced so much more. Now we can turn around and look and say, well, what's going on? How does this vaccine work? And from looking at how does this vaccine work, possibly identifying other approaches that would then be supplemental to the vaccine to actually widen the applicability or the application of treating measles if there's an outbreak.
Yeah, well, I mean, let's just make sure everybody understands that.
Sure. Yeah.
Vaccine debuted in 1963, right? So it wasn't a lot of.
It's a good year, 1963, because I debuted in 1963.
Well, but the fact that it's amazing how highly effective this was, and it isn't until 2026 that someone, has the Sapphire and Dr. Sapphire there has been able to identify the 4 antibodies, right? Through a retrospective study, they've kind of found 4 antibodies that bind to the MMR kind of proteins, right? But it's in what they underlie in the story is because it worked in 63, no one's really questioned it, right, John, until they kind of came along and helped identify them. But the real breakthrough here is that now they think they can have prophylactic antibodies for preventing MMR. And that's important because there's, Canada, the article references how Canada's lost their status of eliminating it. So there's like more than 10 million cases of measles being outbreaked, even though I think people are getting vaccines for it. There's still kind of these breakthrough cases where now these antibodies become potential therapeutics, right? Mm-hmm. So I think that's really, and then will it ever replace the vaccine? I don't think they're arguing that, although...
No, like I say, I think it really is there to help other populations.
Yeah.
And again, it's worth pointing out, this is done in cotton rats, which I've actually not heard of before. But again, it's similar to the previous thing where it's been done in rodents and showing some applicability because let me see. Yeah, so the antibodies were able to halt virus replication in cotton rats, when given either before or as long as 2 days after exposure to measles. And so the 4 antibodies actually have different mechanisms. 2 of the antibodies target measles hemagglutinin surface protein, which the virus uses to attach to cells, while the others bind to the fusion protein used to physically pierce the cell to grant the virus entry. The fusion protein springs forward in an irreversible way, like a spearfishing rod, to change shape and drive itself into your human cell. So that's where the headline comes from about, stopping measles from stabbing cells. So it's interrupting that fusion protein.
Yeah, well, I mean, no, I don't think anybody's really thinking about MMR research or vaccines, but here we are with 10 million people apparently having it. So holy smokes, that's a lot, right? And so,
Yeah,
probably,
actually, the 10 million is in Canada. It's worldwide. There are 10 million cases of measles every year.
yeah, yeah,
Yeah,
which is
so
still high though, given a
yeah,
vaccine that's
absolutely, yeah,
effective.
yeah,
And so
but
I'm guessing somewhere in there they thought, hey, we may, that probably was the activation energy to get people to look at, hey, what are these things doing? And you're absolutely right. There's still caution to be made, but this is hopeful for the next wave of, let's say, prophylactic therapeutics, not necessarily vaccines, but it would be nice if a physician who has someone who's been vaccinated and they end up getting measles, or or even vice, say they don't have it and they get it. There's not a lot of therapies for it. This could be really groundbreaking for it, right? But
yeah,
I do think you're right in that this cotton mouse model is not a disease model, so they kind of call it an infection model. But what I'm driving at is they reference non-human primates and ferrets, and I don't know if you knew that, John, but
yep,
ferrets are great models for any sort of vaccines. A lot of your AAV models early on were being done in ferrets and still are, but just for whatever reason, the ferret immune system is very predictive of human immune systems for vaccines. Where this cotton rat, and never heard of it myself, John, so I don't know anything about that cotton rat, but it seems pretty kind of maybe not the best model as they indicate, but it's more to come. But like it's a really it goes back to what we always like to talk about, and how that those basic research labs are really critical to advancing diseases. And more importantly, if something does kind of come up, there's some lab somewhere with something as a potential therapy, right? And this is a classic example of somebody being ready for what I think there is somewhat of an immediate need, but this has incredible benefit if they can indeed start to identify antibodies that can treat MMR diseases.
yeah,
Great.
yep.
Yeah. Cool
So
story, John.
let's move on.
Yeah.
So it's two down. This one now another topic we revisit regularly. This is protective genes sent to the liver created. Sorry, let me rephrase that. Protective genes sent to the liver cleared brain amyloid in mice, and so this is moving into Alzheimer's, where we think about the presence of misfolded amyloid protein in the brain, but most of our approaches tend to be to penetrate the blood-brain barrier to address, obviously what's going on in the brain. But this study suggests that crossing the blood-brain barrier might not be necessary at all to clear amyloid plaques, and that a one-time gene therapy could one day prove an effective treatment for Alzheimer's. This was done at Army Medical University in Chongqing, China. They had a gene for protective protein was successfully delivered to mice, leading to reduced plaques and cognitive improvements. Unlike other gene therapy approaches, this technique uses a standard liver-targeted vector rather than trying to get into the brain. So it's basically they know that a variant of the apolipoprotein E gene known as ApoE4 is associated with a higher risk of developing Alzheimer's disease. But more recently, protective versions of the gene have been identified, most notably a variant called ApoE3 Christchurch. And this was the potential the Christchurch variant was sort of identified when there was a woman who had copies of a mutated presenilin-1 gene. Mutations in the gene are known to cause early onset Alzheimer's that typically leads to cognitive impairment beginning around age 44. But because she had two copies of the ApoE3 Christchurch, she didn't begin developing symptoms until her 70s. So the researchers basically packaged the gene into a viral vector, injected it into male mice with the risky ApoE4 variant, and so they say ApoE is also expressed in the liver, and that's where the gene therapy went to. Treated mice showed smaller amyloid plaques, less brain inflammation, and performed better in maze memory tests. I will pass it to you because I'm sort of somewhat struggling to digest this.
Yeah, it's so John. I think the gist of it is that you can treat Alzheimer's, which, as we all know, is a neurodegenerative disorder, and we got the blood-brain barrier, which is a real problem. But you can treat it through activating therapeutics in the liver, right? That's what this
Yeah,
has shown. And then the liver somehow, whenever it's producing these protective proteins, it's going to make its way into the brain and reduce the plaques, right? That's the APO. I believe the ApoE3 helps do that. But it's really it's kind of it gets back to just how creative and I don't know if luck's involved, but this basic researchers are doing these things over here that normally we might some of us that have been focused primarily on like drug development and late stage candidates and preclinical studies and clinical studies. This is way upstream of that,
sure.
really that true basic research. And obviously these investigators were incredibly surprised that therapeutic area things that were augmenting the liver were curing Alzheimer's. So
Yeah.
if nothing at all, these types of articles, because it's hard to untwine exactly how this is working because there's so many different forms of paper.
Yeah, and there's an independent, they personally asked for comment, a guy called Jason Ulrich, neurologist at Washington University in St. Louis, who's sort of saying, I need to know more about this mechanism as to has this got potential or not. According to the Chinese researchers, their thought is, and it's all theoretical at this point, the ApoE3 Christchurch clears amyloid out of the blood. This gene variant helps the liver and immune cells called monocytes remove amyloid from the blood, and because the brain and blood maintain an amyloid balance, this leads to an exodus of the disease-causing protein from the brain. That's the theory behind it. So obviously, their next step is go to non-human primates, see if they're seeing the same effect there, which comes back to the previous article we're saying that, obviously mice aren't a great model for people, but non-human primate are much more relevant for us. And so it's a watch this space kind of thing, but it's sort of fascinating that you got potentially going for the liver could have. I mean, it's all about the interchangeability within your body, but it's still, it's still fascinating that this could be a mechanism because we talk a lot about, trying to get across the blood-brain barrier with large molecules and all that kind of stuff, and at least for this particular application, this alternative route may have potential. Who knows? We'll see.
Yeah, I think there's been 15, 20 years of research dedicated to neurodegenerative type of medicines, and there's been very few successes. There are some approvals, but I don't, they're not the most effective at preventing or treating it. And, it's always been difficult because of the blood-brain barrier, because of the different, I think the different, every human's like a snowflake, John, right? So it doesn't matter how, the disease itself doesn't have any etiology that's like a single point. That's why there's
Sure.
been over the years biomarkers like A beta proteins, tau and phospho-tau, and even APOs. So they got all these biomarkers that, and even when they think they've discovered one given biomarker, that if it's elevated, that's going to be indicative. But what they're finding out is no, you can have people at normal levels of that, but there's always these like redundancies and some sort of ratio. I really do think it's like a cocktail of biomarkers that kind of creates the disease itself. And these plaques are nasty. They can, but this, no one, I think this article in and of itself is a demonstration that, man, you got to really think outside the box. I don't think anyone could have predicted that treating one's liver, and it seems like in this case you're arming monocytes to go mop up the ApoE or whatever sort of proteins might be causing the plaques in the brain. But it sounds like the monocytes are getting armed within the liver, and then they're trafficking their way into the brain to kind of become this defense mechanism for preventing it. But this is still very early on, John.
Oh yeah, absolutely.
A lot more to go on, but it's really, I want to hammer home the thought of like just how amazing it is that someday where it's, and by the way, treating somebody with drugs that try to get across the blood-brain barrier is obviously a challenge. They've had a lot of drugs you try to label with something to have for active uptake, but that becomes very problematic again because of that barrier. And then some drugs are given intracranially.
Mm-hmm.
You can imagine how that isn't really a sustainable way to treat people, because if you're in an early stage, the last thing you want to do is maybe tap into someone's brain. But man, if you could just give someone a shot and augment their liver and they can treat Alzheimer's, man, that's a solution, right, John? Like that sounds good.
Yep.
And then I'll round out by, hey, stay tuned on the let's call them in vivo vector-based CAR or even therapies, right? So the AAV space has really got some impact, and not like arming T cells and things like that, not the like the ex vivo autologous therapies, but more the in vivo vector based therapies could be very beneficial in the Alzheimer's space. But I would invest in this, John. That's what I'm thinking. This has high upside if you can start unlocking how the liver, because the liver is a very easy organ to reach and turn on, so to speak. But fascinating stuff, John. So we are we going to do one more?
Yeah, let's move on to the main topic. I think.
Okay.
I think it makes sense. So I'll introduce it and we'll see where it goes from here.
Sure.
So obviously, we've been a business that's been going for over 40 years, and we're slowly expanding our services to meet the needs of the evolving requests from the pharmaceutical industry. So we thought maybe just worth touching on. We're giving it the title "Recent Trends in Bioanalysis: The Evolution of Client Needs, Instrumentation Platforms, and Regulatory Requirements." Because if you look, even if you go into the lab today, the instrumentation is very different from where it was 10, 20 years ago, because the therapies have become more and more complex, and so the analytical methodologies to deal with those has become more and more stringent. And it's not just about complexity; it's we want to go and look at lower levels. The challenges never stop. I mean, we can't sort of rest in our laurels as a bioanalytical lab, because the needs are always evolving. So we always have to do what we can to meet those needs and ensure that we can help in drug development.
Yeah. No, and John, I definitely think that this exciting topic, right? All these, one of the best things about working at a CRO is you do get to have a good pulse on trends, and trends come in the form of what latest and greatest drug modalities, the instrumentation platforms that we get to deal with, regulatory requirements, and in general our client needs, right? So we've both I've been here 13 years at KCAS Bio. Over that, you're rolling up on 5, I think, right? And we've had a nice it's been wonderful to see the evolution, and that brings us maybe even to the first topic here, right? Like we always know that in bioanalysis there's often this need for just expanding the sensitivity, John, right? Now, maybe I'll let you go first, because I think in the mass spec world, even in my time here, I remember when we got our first 5,000 instrument, and that was a big breakthrough. But now you can go ahead and tell us more about how advanced SCIEX, and that's a SCIEX.
Yeah, I mean, obviously we're a SCIEX shop in common with many, and they've had an interesting evolution over the years. They're early instruments with the API 3, which in terms of sensitivity, etc., and this I'm talking going back got 30 odd years, and the API 3 Plus was an excellent instrument in terms of sensitivity, and none of the other manufacturers could match it. Then we moved to 300, 365, which wasn't a great instrument. It gave the other manufacturers a chance. But in terms of SCIEX have done a lot of work in terms of looking at the and the other manufacturers too, is how can you better improve the sampling from the ionization process. So we then went to the 3000, which was a step ahead. Quantum leap from 3000 to 4000. 5000 was an improvement to the 4000 in terms of sensitivity. And then we had another good jump from there was the 5500, which didn't make a great deal of difference, but then 6500 and 6500 plus was another big step up, and the needs of our clients were always pushing for these low levels of drugs in plasma and other matrices. Now we're at the API 7500 and 7500 plus, and it's interesting you talk about you see the API 5000. For us, that's now a platform that is being retired because we need, with particularly with large molecules, you need those higher performing platforms. So we're almost exclusively, our fleet has moved to 6500s and 7500s, because obviously you can redevelop or revalidate and show that assays that worked on a older platform can now be readily run on a newer platform like the 6500 and 7500. But you're never going to, an assay that you have running on those two platforms, you're not going to get running on the 5000s and lower. So although those were great instruments, their time has come, and we're much more in the 6500 and 7500 fleet, because that's where you need to be and it's a combination of things. It's actually looking for the sensitivity that's needed for getting down to low levels of these drugs that we're being asked to investigate. But it's also trying to minimize your consumption of samples. So it's like, for us, I remember the early days, again, when we were at API 3, etc., we were routinely extracting 200 microliters of sample, or half a mil of sample, or more than that. Now we're at 25 to 100, and 100 is a large aliquot to be using. We're much more than that 25 to 50, because the improvement in the instrumentation, it means that you're not consuming as much, so you don't need to take as much. And it also then helps in terms of construction of animal studies. You can maybe take serial samples from animals, as opposed to a lot more sparse sampling. So you involve more animals, and that means more animals is killed at the end of a study, things like that. So that it's a lot interplays. So the sensitivity of the instruments can have a huge amount of benefits to how the whole sort of process works.
Yeah. No, and as a couple things, John, you mentioned our 5000 suite there. We had a bunch of new 7500s and pluses arrived today, and by September our 5000 is going to be gone. I was on site today.
Yes.
5000, they're going to be gone. And then there's even an 8500, right? But that's a high res instrument.
Yeah, right? I think it's the 8600, but
the 8600.
I think it's like I don't know too much about it, because I think it's more of a time of flight based high res, so different from the Orbitraps that we've typically worked with. But again, there's a lot of attention being paid to the 8600 and what it might offer. Because of course you have to keep abreast of the developments and what these instruments can offer you in terms of you got an assay that might be a struggle on one platform, but you come at it from a different angle that you might then have a solution.
No, I think it's just, even as I sit here, I'm fascinating that I've been working here long enough to see. At the time I joined, the 5000 was it was on the market, but it was a big investment for us.
True.
Big step up from the 3000, 4000 instruments, and now here we are. They're being obsoleted because they're not sensitive enough, right? And sure. I think we're of the 40-ish instruments we have, I think we're going to have something like 14 that are 6500, and the 26 of them that are going to be in the 7,500 or higher range. So we're really, I think I hope our audience hears us. We are state of the art when it comes to mass specs these days. But John, maybe switching over to the other, so on your side, you've got the sensitivity
Yep.
with the instruments. And then on the ligand binding assay space, or in general, maybe large molecule biomarker space, the latest and greatest instrument or platform is the NULISA system, which is from a company called Alamar, and they sell an instrument called the Argo. And this is really, you touched on sample volume. One of the first things right out of the gate that makes this instrument incredibly attractive is the ability to use like 25 microliters per sample, and it requires almost no dilution of the sample. And so if you that the ability to use a small quantity and get readouts, they have some plexes that go up, and I think they're launching one in the 350 range. But the technology itself, the advancements of the technology come from, you're still using antibody pairs.
Sure.
And one of the antibodies is biotinylated, but the other one is a poly-A tail. So they're using like PCR for the readout, right? So this is the instrument itself has it's like a qPCR box, and then when you get up into the higher panels, you actually have to load the instrument up and then move it over to a next gen sequencer. But the reason why this gives you this high sensitivity and even specificity and selectivity and everything is it's like an immuno PCR reaction now. So it's kind of, immuno PCR was a technology that's been around for a while, but it never really worked well because it was challenging because you didn't have an instrument or anything. It was you kind of had to hodgepodge your way through it, maybe do an upfront immunoassay and then kind of try to adapt it to some sort of PCR instrument, where the people at Alamar have capitalized on that, and they're taking antibody pairs that normally detect in the nanogram level, and they're getting sub picogram, femtogram, or even down to allomolar ranges of detection. And this has become, it's really taken off for us. We've had it for over a year. The Alamar is really looking to partner with us on a number of their kind of single plexes, some of their homebrew kits. A lot of it's focused on the neurodegenerative space. We're looking at doing some fit for purpose validations of their 5-plex, which is interesting enough. It's got ApoE on it, and things like NFL and tau and brain derived sort of biomarkers as well. But the fact that it can take commercially available antibodies and make this incredibly sensitive method out of it is really, I don't know if it's revolutionizing the biomarker field, but it has huge upside for biomarker analysis, where not only are you not having to do any dilution correction. So if I'm measuring a spattering of biomarkers with a 1-to-4 dilution and a bunch of other at 1-to-10, that can create everything's done at the same dilution. That's huge. Has a very large dynamic range to it, so that you can get detectable readings on just about every sample. So meaning, BQLs don't really help a scientist that much. They've overcome a lot of that.
Sure. Yeah.
And it's a wonderful platform. And then I'll say we're evaluating it for PK, and there was a publication out showing that there was some advantages to doing it by PK, and PK has huge upside because, again, same thing there. If you're doing different doses, right, dose escalation, you almost got to create 2 curves sometimes, right, John, because it's so vastly different.
Sure.
And in this instance, and that dilution itself, I mean, statisticians can overcome some of that, but the data is a lot cleaner if you could use 1 dilution. You don't have to pool animals because you can use low volumes to get the answers. And so stay tuned, but we've seen some sponsor success. We've got a sponsor that we're looking at doing this, and it's showing real promising. Like, we adapted an MSD method, and it was in the mid picogram, like 50 to 100 picogram level, and now we're seeing it well below the picogram detection level, and this is got a sponsor very interested. And then we are working on a generic assay, which, if you're familiar with what I mean by that, it's measuring some sort of monoclonal antibody or any sort of antibody-based conjugate, where you don't need any special reagents, and you get high sensitive methods. And you can, we have generic or universal assays by ligand binding assay, and then we have some by hybrid mass spec, right, John, where you follow the digested peptide. So I think when it comes to like trends in bioanalysis, sensitivity is always going to be on there, John. And as we work here at KCAS Bio, I think we're right on top of it with our investments in mass specs as well as in the NULISA system.
Yeah, absolutely. Yeah. I know. I know. I've been in discussions. Where you talked about some of the Alzheimer's or the neurological biomarkers, and the levels that people were wanting to measure didn't seem feasible. Whereas now you're talking, it's much more
Yeah.
available to you in terms of what the technology is
Well,
giving you.
not having to take CSF, right, John? Plasma
Sure.
measurements that have
Yeah. Sure.
meaningful data, and there's just a huge advancements in being able to not have to sample CSF, but still be able to, and it can be a good diagnostic tool as well, right? To start looking at, and then one of the projects we hopefully will be working on is like we got if you looked at healthies, cognitively impaired and true AD patients, that would be a good thing to be able to discern, to sort of start, the cognitive imperative means people are kind of progressing. But if you get biomarker panels, I mean, I don't know about you, John, it'd be real nice to know. Oh, do I
Yes.
want to know? I don't know. I'm kind of torn by that now, but it would be really good to, if you're predisposed or any of those things, it'd be good to be able to really start to understand. It's always, if you can diagnose a disease earlier, you always have a better prognosis. And that's somewhere where the NULISA could really start to tease out, and not having to take CSF samples, where if I could envision really large longitudinal studies that can help you really understand that, and really arm physicians with taking a plasma sample and being able to help those individuals that are worried about it. Right, John? So anyhow, my 2 cents there.
So there's your high sensitivity piece.
Yeah.
So the other
John, what
side
else you got cooking
other
up?
side of the coin for us is then high selectivity. Again, improvement in the instrumentation
John, did
has
I
made a
lose you?
lot of it made a lot of difference. For us, it's on the mass spec side of things, we're steadily building a fleet of high res mass specs. I've had assays in the past where a triple quadrupole just could not filter out enough background to allow you to measure your analyte of interest. We touch more on oligonucleotides later, but we found that again, the having high res instruments for oligo is just it helps in terms of reducing development time. Yeah, you're cutting out background because that's really where the selectivity becomes key. Your sensitivity, you actually gain sensitivity from the selectivity because your signal to noise is low, and so we can tease out data from from background. The having that wide range of platforms between the triple quads and the high res mass spec is really it greatly enhances the suite of what you can do for your customers. So you need that whole set of platforms to really address the assay needs.
Yeah. And John, so on the ligand binding assay side, we have a variety of platforms that are highly selective, but one that's somewhat new to us, although I think it's been around a while, is the alpha ELISA. And it's a little underutilized, I think, but we are finding more and more sponsors that run it in-house because it has great selectivity to it. And how it works is, you just, again, it always starts with the critical reagents, all those are near and dear to me, but you
Yep.
got an antibody pair. And in a conventional ELISA, you might capture with something and then detect with the other, and then in an MSD format or even Samoas, and you end up using some sort of biotin, kind of streptavidin, kind of helps get more capture antibody, better detection there. But on the alpha ELISA, it's all in liquid phase, and what they use is they call it like an alpha donor bead, and you biotinylate the antibody onto this bead, but this bead, when you hit it with a laser, you can get excited. And
Mm-hmm.
then they have this alpha ELISA acceptor bead, and on that is the technically the detecting antibody. And so when the two of them get that when they bind to each other and you get the analyte of interest and you've got the capture antibody and bead on there and the detection antibody and bead on there, you hit it with a 680 absorption or laser, and then what it does is, because of that proximity, it emits on that like acceptor bead, and it gives you great signal. They're tough methods to optimize, John, and it's a service that what we're finding out is I don't want to call it an oldie but goodie, because I do think alpha ELISA technology has been around for a while. It's something we've been offering for almost a year and a half or 2 years now. But I do think it might not have quite the sexiness of a high-res mass spec and some of what's going on over there. But I think it's important to mention how this is, as we begin to think about trends in bioanalysis, the evolution of our clients' needs, as well as like the instruments and things that we need, this becomes it's actually another tool in our tool chest. And I might not start with an alpha ELISA program, but to those that are out there using alpha ELISAs that need CROs, we're now got a couple of these under our belt, and we really feel like it's something we can help you out with. But it also could be when you're at your last straw, it's another platform to go and look at to provide good quality data. So I don't know, John, if you have anything to add, but
I think it's that whole thing. Yeah, we talk about high res mass spec being sexy or whatever, but ultimately you just want as many different tools available, because where one might fall down, you want to have something else that might be able to step in and take on your problem and solve it. So yeah, it's like the high res is obviously, that's your big investment instrument at the moment, but again, that may change with time, as we see with all mass specs anyway. They're very expensive on initial launch, but as they become more accepted and the cost goes down, but who knows what might be the next improvement in mass spectrometry quantitation downstream from here. So we talked selectivity, we talked sensitivity. As we talked about trends in bioanalysis, this is something I don't think about at all. But there's a lot of discussion in this on your side of the business in terms of single replicate analysis. And I remember, being at EBF, it must be, several years ago, and it was like afternoon workshops. And, when you're at a conference, you like to sort of dip your toe in other areas and see what has been spoken about. And this was my first inkling about, the norm for multi-replicate analysis within ligand binding assays, where for us it's just you do one measurement and that's it. So I'll pass it back to you, because I just want to at least give a bit of my background to it, and like I say, it's not something that affects me, but I know on the like I said, ligand binding side, it's a hot topic.
Yeah, no, John, it's fascinating that you've never had any replicate sort of issues on the mass spec side. I guess I kind of knew that, but you reminded me that. And in our world, I've been doing, I feel like I've been doing this so long. Everything was triplicate at one point.
Yeah.
Right. And then even as I got into the CRO space some 18 years ago, we almost never run triplicates because of cost, right? It was always just duplicates. And then there are still some methods where it makes sense to maybe prove that you can go from some cell-based things or activity assays. You might even do 4 replicates and then triplicate and down to duplicate. But the norm is duplicate, and you touched on it. It was actually, I think 2 years ago at EBF, it was a big section of the afternoon. And so, the PCR space, I'll say that they've been doing singlet for a long time too, John, right? They don't do a lot of replicates there. And those are even like, IVD approved tests that are a single rep, and it's pretty, physicians rely on it. But it's been kind of like taboo in the LBA space, just because of like the I don't know, the stress when I was an analyst, I would have hated running singlets. It just gives you more confidence to be able to do duplicates. And I hope you can kind of hear it in me, John. I think if you had this discussion with me, maybe even after EBF, I wasn't sold after them, but the field is really pushing this.
Yeah.
I'm kind of warming up to it. I hope I don't sound like I'm stodgy or too conservative or anything like that. It's just, I try to look at all angles, right? Not just saving on money and things like that, because although I would say that's going from duplicate to singlet isn't going to save you 50%, John, right? It's going to have you're going to get some savings, and we put it at it. We are offering it now regularly. We have some projects where we're doing singlet replicates, and it's maybe like a 20 to 30% sieving, which is significant, and it also reduces footprint, right? Using less materials, all good things. But I think I still want to put a little bit of caution around this, in that you really got to prove that the singlet replicates are there.
Yeah, I think for us as bioanalytical scientists, you want to do the right things. You don't want to jump to an alternative approach until you've really proven that it is safe.
Yeah, I think,
for want of a better word.
Yeah, well, I think there are some people who are, maybe a little bit more risky, and they're convinced right out of the gate that singlet can be done. But I think in terms of regulatory submissions, I think if you can prove it during development and validation, and you want to go into your sample analysis at singlet, I think you'd be okay. But again, you got to demonstrate it. It's not just like I'm doing my development and validation in single replicates and calling it good. You still run across, there's an argument that if you do singlet for bioanalysis or sample analysis, and you get a result that's just spurious, failed, whatever it might be, do you have to rerun the whole plate, John? That becomes the issue there. And then you can counter it and say, well, if you were running duplicate replicates, there's a good chance even if one of the two were out, you still got to repeat that. But in my world, if you got two of them, if you can do a grub test on there, you can kind of confirm through stats that one of them was plate washer got blocked, the technician had an error, something like that. But the world seems to be pretty convinced that you can do singlet, and we'll have some case studies out here soon, John. But I do feel like it's going to be the wave of ligand binding assays. But I think it's going to be still going to be somewhat method dependent. I don't know how quickly it will adapt to PK and ADA, but in the biomarker space for exploratory biomarkers, I think it's really becoming more the default would be good. It's not there yet, but I think in time it will. And then I think slowly but surely you'll see it in the PK and ADA space. But it's somewhat new to us here, John. It's not something we would routinely be offering, even though we talked about things from a couple of years ago. But we want to do as you said, we want to do what's best for our clients as well as the individuals doing the work, right? There's a little stress there when you go down to singlet. But I think you get confidence, right? As soon as if you're an analyst and you're trained on the method, you should be fine with running it in singlet replicates. But it is a hot topic in bioanalysis.
So do you routinely do ISR, in cord sample reproducibility?
Yep.
As part of, so you're doing that because again that's our defense for showing that singlet is fine, because obviously when I started we weren't doing ISR and there was a lot of discussion back and forward, and then it became it became the norm for us to be doing in cord sample reproducibility in talks and clinical studies, and I obviously I've had assays that they weren't functioning well, and we would never have known unless we did the the ISR that pointed out that there was a problem, and it has made, in my opinion, it's made things much more robust. So so although you're doing it, that you still would like to do the duplicate.
Well, yeah. So you bring up a good point about PK. There's no ISRs for ADA and biomarker.
Okay, yeah, sure, absolutely.
But for PK, if you do have a result that is an outlier, maybe a mistake, whatever it might be, you can come back and retest that as part of the ISR batch. You're absolutely right. But in the, if I'm doing a biomarker 220 plex, man, and I got one of those wells that I'm questioning, I don't know. That's a very expensive retest. So, but there's ways to, it's hard to address exactly what you do there. Although I think if you've got the rest of the time points, you can maybe go without it, or there's some workarounds. But you bring up a really good point about ISRs reinforcing the PK side of it, but they're not done.
Sure.
But there are some people who want to do them. For biomarkers, there's a lot of resistance from many people, including myself, that would tell you there's not a lot of value in doing ISRs on ADAs, because you are doing the 3 tiers. Although if we're going to 1 tier, maybe there might be. And then the biomarker space, there is like a small group that's always opining about wanting to do they don't want to call it an ISR because we don't truly have like a standard and things like that.
Sure.
It's just more like an incurred reanalysis.
It does become, but it's a reproducibility of measurement, so long as because your sample's a snapshot in time, you want to know that snapshot is consistent anytime you go back to it.
Some LTS does some of that. I don't know, some arguments around how to do that, but there's, I'm certainly not here to promote one way or the other when it comes to doing ISRs on biomarkers and things like that. But I like what you said about reinforcing the PK data through ISR is a great way to kind of check yourself, make sure you got it doing it right.
Yeah, and when you talk about measurement of 220 biomarkers, that's not something I ever have to worry about on the mass spec side of things. It's not very often we're going for many analytes. Anyway,
that's the trend. That's another trend there. People are, biomarker high throughput panels. They're not designed to test thousands of samples on.
Sure. Yeah,
but there are people who are starting to look at it the other way around, which is interesting. I don't know. That makes sense. Some people they're coming to us and they're like, "Hey, we got 500 samples. We want to do a retrospective study. Can we do them all on this, 220 plex or whatnot?" And we're kind of like, it's better to maybe take a pilot sample, take a spattering of them, some sets of them, take a look at, filling a plate up, and then go from there, see if it's worthy of it.
Yeah.
And even then, after you get some hits, I would say, why am I testing 220 analytes? Maybe there's only 5, 7, 10 in there
that are relevant.
I would want to run my other 500 or 5,000 samples on. So there is some strategy involved there when it comes to maybe biomarkers in general. But okay, John, we're moving right along, right? We got a couple more topics we want to touch on.
Sure,
certainly near and dear to me.
This is mainly you because I think that's. Well, I suppose it's both of it. It's like this is again a lot of discussion in the bioanalytical field, and a lot of this was again EBF in Europe have really been driving this is the use of fit for purpose validations and biomarker assays. I mean, I've sat in a lot of sessions regarding this in Barcelona, and so I mean it, and actually we're seeing it more in terms of talking to clients. That it used to be that we get on calls and they'd say we want to validate this biomarker assay, and it's like, but do you really need to? And there'd be a lot of to and fro. And I find that everyone's much more calibrated in terms of what their expectation is, and they are just doing a precision accuracy run, limited stability, whatever. And it was interesting to have a call earlier this week that we were both in, where it was these two biomarkers are absolutely a primary data point that we need. These have to be validated, but that's an unusual request nowadays.
Mm-hmm. Yeah, no. So it's a little bit to unpack, right? Because a lot of people use terms like intended use, context of use, fit for purpose, and I think sometimes people just want to say, if you slap a fit for purpose validation, you can do whatever you want. That's just not a true statement, John.
No,
I think I want to be making sure that we understand that, when you do a fit for purpose validation can have many, many flavors to it, but it still has to be driven by what are we doing with the data? What do we have that we can do to make the method robust, specific, sensitive enough? So there's a lot of things that go into play when you talk about intended use and context of use, and that's a whole separate podcast.
Sure, absolutely. Yep.
But I think for what I'll say about this, why is it such a trend suddenly for fit for purpose validation? It's because there's a war on the word qualification, John, right? I think it's created a lot of confusion because I feel like back in 2010, there's some guidelines that came out that made it very clear around what your fit for purpose was, but they never talked about qualification versus validation. So what's tripping up our industries is, in our world, John, our bioanalytical world, if someone said they've got a qualified method, you and I don't think that it's a bad method that couldn't meet performance characteristics. We just assume, hey, the use of this was, they didn't need a QA review. Qualification was good enough for their uses. But if you're a CMC person, if you're running a qualified method, that means it failed validation, John.
Oh, really? Okay.
Yeah, that's where the confusion lies. So when you've got people who are If I say to someone, oh, hey, we need a validated method, but if I'm talking to somebody who might be more of a CMC GMP expert, who's now maybe finds themselves needing to do bioanalysis, if I say, hey, we're going to qualify the method, that they take that as like, oh my God, what happened? Oh my God, it's gone disastrously wrong.
Sure.
Because in the CMC space or GMP space, that's how they define something. When you're qualifying a method, it means that it failed validation. And by the way, it still gets a QA review. Now, in our world, the term qualified means it lacks the QA, but everything we do is done in a GLP environment. It's still going to be done in a similar fashion. It's just not having that back end QA review. So now,
and it's also, it's basically showing that the assay as it's developed is suitable for use to meet the needs of what the client wants for that particular study.
Yeah.
That's where we're going with qualification.
Yeah, I mean, I think to me, a qualified method in our world could have percent CVs, percent recovery, all less than 10 single digit levels, would fly through a validation. It just didn't, because of the intended use for what and by the way, you can submit data and any sort of submission to a regulatory body that doesn't go through a QA review if you've a priori your data that way, right? So that you've set it up the right way. But back to the way we're kind of looking at it now, because the world is, I think there's a lot of members of EBF and there's a lot of members of the APS community and even the WRIB communities where they're just like, okay, we're going to call everything fit for purpose validated, but nobody's really addressed that regulatory rigor. So I think I still think this is
still in flux,
something that might trip us up. So we're starting to talk about it in terms of like a regulated fit for purpose validation or a non-regulated fit for purpose validation. But some people don't like that, because if it's fit for its purpose, and that purpose is 3 A&P runs with no qualification, no plan, no report, right? No, sorry, 3 runs, no formal validation report, no formal validation plan, and no QA review, that can still be a fit for purpose validated method.
Yep.
But that gets hard. There's a lot. It's going to create a lot of confusion. And what the last thing we want to do is be deceptive and say, hey, we validated this. This is a fit for purpose validation, and all it is one A and P batch, John. Right.
Yep.
So that and then we've started thinking about tiers. If we do have a bunch of tiers, but there's a lot of people that are. I'm starting to hear about, oh, these tiers are silly and this and that. Well, if all I have to say to some people is, if you keep hating on things, please come up with a solution. Because just telling me that everything can be fit for purpose validated and it's a blank canvas, that right back to where I started with. Don't just slap a fit for purpose title and do whatever you want. That's bad science.
You have to have agreement between you and your customer that what you're suggesting for that fit for purpose validation makes sense to both us and them.
Well, I mean, even think about the reviewer, right? How about the person who's reviewing the submission? If you just put fit for purpose validation on it and you don't put any caveats around that, and then I start looking at your data and all you did was 1 A&P run, I'm going to think that they know what they're doing. But then maybe you have a fit for purpose validation that's got 6 A&P runs. You put a plan report and it's got, 180 pages document for its validation report, but you're going to call it the same title. So I still think there's some things to be ironed out there. And by the way, I'm involved in a whole bunch of communities where we're trying to hash this stuff out. And the problem with that is it's an echo chamber. And there's maybe within it if we just let a small community of people dictate things, it's going to be problematic, because there's a whole bunch of people. John, remember, if you're talking to a European client in general, if you're in a bioanalysis setting and you use the word qualification, they're going to think that you don't know what you're doing. But regularly here, we take calls all the time from people that are very, very good in translational translational sciences that say, hey, I need a qualified method. So it's still to be determined on just where we're going to land. Although there was a publication back in March that said you should avoid using the word qualification.
We'll come full circle at a later date, probably.
So, all right, I think I beat that one to a dead horse. But I got opinions, John. I got opinions on
Let's move on. Final topic, because I think this really this is indicative of why having this suite of instrumentation available is key, and it's what we're seeing. I mean, probably, I always, as a mass spec person, we had very little contact with oligonucleotides. I think the first time I ever worked with any was actually on the time of flight, my days at Kratos. But when it came to a bioanalytical lab, we there was no real discussion of LCMS, of oligos, because they're tough analytes. There's a lot of problems with absorption, carryover, all sorts of chromatography could be temperamental. Just a lot of reasons why you know we weren't getting any requests, as does be more and more emphasis on oligonucleotides, both in terms of just straight oligos or siRNAs, or ARCs, where you got the antibody with an siRNA. The mass spec has become more integral it's a more integral tool with everything else that was already being used in terms of giving answers for these oligonucleotide type of analytes. Again, for us, we're having that full toolbox of triples and high res instruments is vital to in terms of meeting the needs of oligonucleotide analysis. And even I was having a discussion yesterday with Ravna about what they do on triples, and although the API 6500 is a lesser sensitivity instrument than the API 7500, what they find for some oligonucleotides is, because the signal to noise on the 6500 is less than the 7500 because of the smaller orifice up front, they can actually get better performance for some oligos versus 7500, and then they can get obviously a complementary performance on the high res instrument, so that it's that variety of tools. But I didn't want to get too far down the mass spec side. I'll pass it back to you, because we're using a lot of tools for oligo measurement.
Yeah, it just goes to show you can't be absolute when you're talking about the sensitivity of each of those instruments. You got to be careful because there are those caveats, right? But in terms of oligos, one of the things that I love working here, many reasons, but we got many tools, John. Right? You've got mass spec, you've got high res mass spec if needed. We have hybrid ELISA, and I'll touch on those in a minute.
Yep.
Very powerful tools. PCR is the fourth one that we offer. And then there's like a hybridization LCMS, right? Which we haven't quite tried yet, but I think that's in there.
We're sort of dipping our toes in on that. Yes.
Yeah. So we have those four buckets, but John, maybe not surprising at all, the PCR is the most sensitive, right?
Yep.
So actually a client, a prospective client asked us, after we went over we have a slide showing all four and the ranges, and I don't have it memorized what the ranges are, but they asked, hey, what would you recommend to start with? And that's a tough one too, John, because you got to kind of, if you got to use the tools that you have. Not everybody's got all those things like we have, sure. Right? So that, to me, it's like I would start with whatever I've got that gives me the answer I need. And putting that aside, right? If you don't, so maybe you can do it by mass spec, but we do have some case studies that show both the hybrid ELISA and the PCR can get maybe 2 to 5 log more sensitive than whatever we were doing by mass spec. We weren't using high res at the time. So to me, it's like the real, the way I answer this is if you came to us today, we would probably default to a PCR reaction, John, unless there was some other reason. Or we would look at what are you more accustomed to that might be beneficial. But the devil's in the details around how sensitive you need to be at.
Yes.
But then it's challenging because are you talking about some sort of arc, right? Where there are huge advantages to doing that by mass spec because you're able to do a 1.5 plex, or I think we've got a 1 3/4 plex, right? We got some terms, but that becomes an advantage. But I do think when it comes to oligos, it's great to have so many tools at one's disposal, and PCR is a tried and true platform for it. I'll say about hybrid ELISA, this has come a long way in the 3 to 4 years that we've been offering it. And what I mean by that is, so you still got to make the same probes. Doesn't matter the platform. You still got to go and make a probe, whether it's PCR, high res, right? You still got to go out and make some sort of
Sure.
nucleotide probe, right? So that's still a critical reagent that all of the platforms need. But the hybrid ELISA, because of some of the more commercially available reagents with the DIG detector is what's used there, the ligation sort of reagents that are used, we're finding that over the last few years, we're getting much more success because people are doing more and more of it. So like any capitalistic market, there are more and more vendors providing it, and the quality of those reagents has improved. So hybrid ELISA is a really good platform.
And I think that's a key point, and I've returned to this multiple times. We're lucky that as end users we have, suppliers on the mass spec side of things, and who are always looking for improvement too. So for oligos, I talked about temperamental chromatography, but the column technology is improving, where they're actually developing phases to specifically deal with these type of analytes. And so, we're really benefiting from a very sort of, I mean, obviously it's commercially driven, but a sort of collaborative supplier set who are looking for tools that will help us and in turn help our customers.
Yeah, no, definitely. And then one more thought that flew into my head is if there's any metabolites of your
Yes.
oligo, then mass spec becomes very attractive. If you
And that,
need the
and
activity of each of them, that becomes huge.
and for high res, that high res really steps into its own on that.
Yeah. So
Because we can do full scan acquisition, and you can actually then go back and interrogate that data later to look for metabolites, etc. So yes, mass spec can certainly do a lot
well,
in terms
but
of
it's
that piece.
only if you need it, right? Because that was the
Yes,
other factor
yes,
that I want to be clear about when we're starting to assess
yeah,
what the preferred
absolutely.
platform is. If you know right out of the gate that you got a metabolite that you need to deal with that has some activity to it, then you just got to go right to the more high res or mass spec, right? Um, but if not, then I still think PCR. Although I think PCR has some ability to do that too if you design it the right way. Okay, John, I think we're wrapping up, right?
I think we probably are. I think I think we've talked a lot on this. Wasn't quite sure where it would go, but this has been quite satisfying. So yes, I think let's like I say, it's always fun to see the developments and what we have to do to address them, and no doubt the challenges will keep coming.
Yeah, no, it'll. The moment you think you got a grasp on it, it'll change, John. I could say if you don't like constant changing and so forth, then the CRO space isn't the right one for. Actually, I don't know if drug development in general is, but it is. It's wonderful to, think about what we just discussed right now. A year or two ago would have sounded very differently,
Yeah,
right?
yeah,
Just
absolutely.
given so much
Yes,
that's been out there.
yes.
But let's do some quick. We got weekend plans going on, John.
Nothing much. I got a weekend trip which I have to do. I sort of probably end up. I'm going to be in Boston. I'm going to link up with my eldest son, but I have other things I have to do around that. Yeah, it's just
But is
not
that
much in
Fourth
the
of July?
weekend. But,
Sorry.
sorry.
Is that part? Because really, you got any 4th of July plans? Are you going to
Well,
what's your?
see, 4th of July. I mean, I'm quite happy to take the time off, but ultimately, I'm the reason you're having 4th of July. So it's not something we particularly register. It's an American holiday, and I'm British,
I didn't
so
know.
yeah. So
No.
I love having the time off. It's like, but July for us is a big month because it's like there's multiple members of the family have birthdays in July, and then I'm actually going back to the UK late in July, and I've got much more planned for that.
Fun.
To got a music festival in Leeds for a day. There's a site called Great British Car Journey where you can pay to drive like old cars from the 50s, 60s, and 70s, which
Oh,
I'm doing. And so that's what, and then it's just going to some of. I've got a few extra days just visiting other small towns. I'm staying in Yorkshire. I'm hoping I can fit in the Keighley and Worth Valley Railway, which is an old steam. They they basically do trips between two towns, and it's either steam train or a diesel, depending
Oh
on the
wow.
timing. So there's like lots of little things I'm trying to do. It's just a nice trip, but it was actually the music festival was what triggered it. It's a band called Sunshine Underground, who I saw their last concert in Leeds several years ago, and that was supposedly them done. And then they announced they're doing this 1 day music festival in Leeds, and they're supported by Future Heads, White Lies, Hard-Fi, and another band who I haven't heard of. But there was enough bands there that I hadn't seen, and I'd quite like to see them. I thought, I'm going to do that. So that's why I'm back in England in late July.
All great stuff, John. I mean,
Yes.
we couldn't be further different from what we think of as vacations, but that's okay. I love that you're doing like what a nostalgic trip, driving old cars and taking steam trains and then going to
Yeah.
watch
Well, I mean,
shows
you
of,
got things like so I've got a couple of days in York, which is a fantastic city. If you ever go to the UK, always do York. But you can actually in York, you can walk the city walls because it still has intact walls round the city,
yeah.
and you can, they've got a few gates round and you go up the stairs on the gates, get on the wall, and just wander around. And York Minster is just a fantastic cathedral, and so yeah, it's just a lovely, lovely city. So it's just I haven't been there for a long time, so it's just been nice to catch up on some of the stuff.
Yeah, and I don't know, John, you've been working with me long enough to know I like to try to blow up the state of Nebraska with my
Sure,
4th of
absolutely,
July fireworks, but
yeah.
we got a curveball thrown at us as, traditions are wonderful, but my son has decided to depart. We're not going to spend the 4th of July with him, which is fine. He's a grown man these days. He's 20, but he's going down to the Ozarks for some fun and sun with the boating and the good time down there. We will go to Nebraska, but won't be a little different in terms of not having him to help light the fireworks off. We'll see just how big the display is this year, but it'll still be a fun time. I do enjoy. It's a lot of fun, and then we do have a little bit of a family reunion with her, and one of them lives on a little cove, so we'll do some water skiing and tubing and stuff like that. So that'll be fun. We'll see if I get up on water skis. It's been a few years, but I do like water skiing, and certainly tubing's real easy, John. That's not hard at all. But we'll see if I want to. Water skiing is a young man's sport, John. I don't know if you've ever done it, but it's for young people.
No. I to be honest, I'm terrified of water.
No.
And so, yeah, you're not going to get me. I'm quite happy going a boat, but anything that takes me outside of the safety of a boat, I'm not.
No. I love the ocean.
It's not going to happen. Oh, I love the ocean. That's. I'm just.
You just
water.
don't want to be in it.
Or deep water freaks me out. So yes. Yes.
Well, all right, John. I think that wraps it up, and safe travels to you, and we'll see you.
Absolutely, yeah. Have a great 4th of July. I was not trying to be down about it at all. It's just like I say, it's one of those things that I appreciate other people's traditions. It's just we have different traditions, different upbringings. We look at things different ways. But I hope you have a blast.
I don't know how we've gone this long without mentioning the World Cup, which is incredibly exciting here in Kansas City. We got our last game tomorrow night, and then there's a good chance that if Portugal and Argentina match up in one of the knockout rounds here, that'd be here in Kansas City. So that's got us a buzz. But the World Cup's been pretty enjoyable. I think it's already set a record for attendance.
We're sad to see Scotland on the brink of going out because I don't think they're going to make it. And
they had a tough draw, John. But
absolutely, that group was horrible, and so I'll sort of switch allegiance to England.
They had a nice win. They've absolutely. I don't know if you know the story about when they flew over into Boston. They wiped the plane out of beer. They're wiping the Boston bars out of beer. They're wiping the Miami bars out of beer. You Scots are taking it serious, man. It's funny.
Well, I just look on and say, well, I can choose to be a Scot occasionally, but ultimately I'm English. But when it comes to the World Cup, because when they qualified, we were back in Scotland last November, and everyone was so excited about qualifying. So even Kath, my wife, she isn't interested in football at all, but she's been insisting on watching the football games or the Scottish matches because she was there and got sort of whipped up in the Scottish fervour at qualifications. Yes,
Well,
I'll switch to England because that's who I more typically
yeah,
follow.
they struggled
But,
in their last game though,
yeah,
but they look
exactly.
they have a potential to go far. And
Yeah.
if you really want to see something funny, go look at the 20,000 Dutch that were doing their little
Yes.
dance in the streets of Kansas City. I don't know if you saw that. There were 20,000 of them doing some
Yes,
little
yes.
moving back and forth and stuff, and it was wild. It's got a nice buzz here. Kansas City's really benefiting.
Good.
But okay, John, take care. Have a great vacation.